Welcome to our today webinar about dialysis. I would like to give you first a short overview about how dialysis works before I talk about advantages and disadvantages of standard dialysis tubings and ready-to-use dialysis devices. First of all, how does dialysis work? Dialysis is based on the diffusion. That means in this case a diffusion through a semi-permeable membrane, and this membrane has a certain pore size, so some smaller molecules will pass through the membrane and the larger molecules will be retained. And the diffusion goes from the place with the high concentration of the low molecular components to the lower concentration of the low molecular components on the outside of the membrane. The selectivity, that means which molecules will be retained and which will be dialyzed, is determined by the pore size of the membrane, and the endpoint of the method is reached when you have a concentration equilibrium. You can start dialysis again after reaching the equilibrium by exchanging the buffer, because then you pull away the small molecular components which are diffused from the membrane to the outside, and you can start again and reach a very good separation of small molecules from larger molecules by this easy method. The applications for dialysis are very great, because you can use that for desalting and rebuffering. You can use it for pH change, so that you can adjust your conditions for downstream applications. You can remove small molecules like dyes or detergents or other reducing agents and other components which might interfere with downstream applications like mass spectrometry or chromatography. You can use it for renaturation and denaturation of proteins. For example, renaturation can be used to restore enzymatic activity of trypsin or other enzymes. You can use it for more special applications like protein concentration. In this case, you use hygroscopic macromolecules like polyethylene glycol, and you pull out the water from the membrane, so then you concentrate inside the protein solution. Or you can use it for binding studies or for fractionation of different sizes of proteins or nucleic acids or other macromolecules, and for more specialized version like electroelution. In this case, you extract nucleic acid fragments or proteins from a gel matrix like polyacrylamide gels or agarose gels, and in one easy step you can purify your proteins or nucleic acids to a high step. The advantages of this method is that it's a very gentle method, it's very easy to operate, you don't need any sophisticated lab equipment like ultracentrifuge or other centrifuges or pressure or vacuum devices. You just need some, like a magnetic stirrer. It is very easy to adjust to a wide range of sample volumes, from small to middle to very large volumes, and you have a wide range available of membranes with different pore sizes. You can use it, like I already mentioned, for fractionating, for example, small protein molecules from larger protein molecules. But of course, if you need high selectivity and specificity, you should switch to more sophisticated applications like ion exchange chromatography or gel filtration or other chromatography methods. You have a minimal loss of sample, because usually the membranes used are very low binding, and compared to ultrafiltration or gel filtration methods, you have minimal membrane fouling. Many factors affect the rate of dialysis by affecting the diffusion speed of the molecules. One important factor is the concentration of the sample and the concentration gradient. if the substance you want to dialyze and to want to diffuse out of the membrane, it has a high concentration in your sample, the diffusion rate for this substance is much higher, because the probability that the molecules will be in contact with the membrane and then diffuse or migrate through the pore outside of the membrane is much higher. A high concentration gradient is as well a good force for diffusion speed. And how do you get a high concentration gradient? It's very important to have a high buffer to sample volume ratio. The buffer to sample volume ratio should be at least 100 to 1. After reaching the equilibrium, you can also start again the concentration, the diffusion, by building up a new concentration gradient, because if you exchange the buffer, you pull away the small molecules which are dialyzed, and you have again a concentration gradient. And the other thing is that you reduce the concentration of the substance you want to remove by dialysis according to the factor, how often you exchange the buffer and how high the volume is of buffer you dialyze against. For example, if you have 1 milliliter sample and you dialyze it against 200 milliliter dialysis buffer, in one step after reaching the equilibrium, you already decrease the concentration of the substance you want to dialyze for a factor of 200. If you exchange the buffer 2 more times, we are talking already of a factor of 8 times 10 to the 6. That means if you start with 100 millimolar DTT, you reduce the concentration from DTT after 3 steps to 12.5 nanomolar. The size and the shape of your molecules which are dialyzed is very important for diffusion speed, and the interaction with of the molecules with the solvent, and of course the viscosity of the solvent, because it's a thermal movement which drives the diffusion. If you have a very high viscosity solvent, you will of course have a reduced diffusion rate compared to more aqueous non-viscous solutions. Why should you steer or shake your dialysis membranes to increase the diffusion rate? Because during the dialysis, the low molecular components wander through the pore size out of the membrane, and during time you will build up a high concentration layer of this dialyzed substances at the membrane side, and of course this will slow down significantly the diffusion rate, because you decrease the concentration gradient. So if you steer or shake, you break up this layer, and then you maintain the concentration differentiation level you need to have a good diffusion rate. Because diffusion bases on a random thermal movement of molecules, the diffusion rate will go up, of course, as well with higher temperature. This is limited, of course, because of the stability of the biological substances you use. So practically, you make dialysis at room temperature, and sometimes you have to make it even at 4 degrees if you have long steps or more sensitive substances, and then you have to live with longer dialysis rates. The diffusion speed is inversely proportional to the size of the molecules, because if you have a bigger molecule size, then this bigger molecule will more slowly move through your solution, and so it will have a slower diffusion rate than significantly smaller molecules. So it's not very easy to diffuse them out of the membrane compared to smaller molecules. Another very important factor is the membrane properties, especially the membrane surface area, the thickness, and the pore size. To have a very good diffusion speed, you have to have a high surface area to volume ratio, because if you have a very high membrane surface area to volume ratio, you have a very short length of diffusion pathway, and this means of course that you have a high diffusion speed. If you use dialyzed tubings, this is the function of the flat width of the membrane of the tubings. If you have two equal length pieces of dialyzed tubings, the one with the smaller flat width has a shorter diffusion pathway, because the flat width, with the smaller flat width, it has a higher surface area to volume ratio, and that means that you have a higher diffusion speed. So this is as well one aspect you should keep in mind if you choose your dialyzed tubings according to volume and flat width. The diffusion speed will increase with the pore size and decrease with the thickness of the membrane. Practically, you can't make too thin membranes because the structural integrity of the membrane will be impaired. So usually the membranes you use have a thickness of around 12 to 30 micrometers, which will give you good diffusion rates and still a very good structural integrity of the membrane. How to choose the right molecular weight cutoff. The average or maximum pore size of a membrane determines which molecules will pass through the pore and which molecules will be retained in the membrane. And this is defined by the molecular weight cutoff, or MWCO, of the membrane. That means that the molecular weight cutoff of the membrane is the size of a molecular weight of the globular substance from which at least 90% will be retained in the membrane during dialysis overnight. On the other hand, that means that you always will have certain loss of bigger molecules during dialysis. There is never a 100% retainment of the substance in the membrane. Besides the size, as well, the molecular shape, the degree of hydration, the ionic charge, and the polarity of the substance will influence how the permeability through the membrane will be influenced. That means if you have, for example, a more longer shaped, a flat shaped protein and not a globular protein, even if they have identical molecular weight, they will have a different permeability through the pores of the membranes. So it could be that a longer shaped protein molecule or other molecules will pass through the membrane, even if the globular substance of the same molecular weight will be retained in the membrane. So there are two topics you have to keep in mind if you choose the molecular weight cutoff of the membrane. First, the molecular weight cutoff should be half the size of the molecular weight of the species which should be retained, and it should be at least twice the size of the molecular weight of the species you intend to pass through. That means practically, if you have a protein of about 12 kilodalton, you should choose an MWCO of about 6 kilodalton, and the things you want to dialyze out of the membranes should be not bigger than about 3 kilodalton. You can use as a dialysis buffer all common biochemical buffer solutions like PBS, TBS, amino acid buffers, or as well water. But if you use water, you should keep in mind that you have to avoid large differences in osmotic pressure. Because if you have a large difference in osmotic pressure from the inside and the outside of the membrane, the water will move across the membrane into the membrane. And that means not only that you get significant sample dilution, the other thing is that you could damage your membrane, or it could even rupture the membrane. So if you, this is of course very important if you have, for example, high percentage sucrose solutions or solutions with high salt concentrations. And if you have, for example, a solution with high salt concentration, you should not directly dialyze in the first step against water, but first use a buffer which contains a lower concentration of the salt, and then go in more steps until you reach water as a dialyzer, because then you reduce the osmotic pressure. For many decades, there are already the standard dialyzing tubings available, which I'm sure everyone knows, and they are primarily made from regenerated cellulose. Since many years are as well available standard dialysis tubings made from cellulose ester. They have the advantage that they have more concise molecular weight cutoff, because the regenerated cellulose have a not very defined and have a very inconsistent molecular weight pore sizes. So the molecular weight cutoff of regenerated cellulose is only an average value. But the cellulose ester membranes have the disadvantage that they don't they are not very stable with high temperatures, and they are not stable with strong acids and bases and many solvents. You have your standard dialyzed tubings for routine applications, which are usually provided as dry format types. You can also use heavy metal-free pre-wetted versions for more sensitive applications, and you can choose between a very broad range of molecular weight cutoff and of flat widths, which makes the standard dialyzed tubing systems very versatile. And this is, of course, one of the advantages of using the dialyzed tubings. You can easily adjust them to many different volumes. You have a broad range of molecular weight cutoffs and flat widths available, and they are very inexpensive and have a long shelf life. But they have as well a lot of disadvantages. One thing is that they need a more elaborate pre-treatment, because for the standard dialyzed tubings for conservation and for more to keep them away sorry, I start again. So you have to treat them with glycerol and with preservatives like sodium acid to prevent them to get brittle. And so you have at least to rinse them with water to remove the glycerol and preservatives for at least 30 minutes. And if you have more sensitive applications, you even have to boil them in sodium hydrogen carbonate and EDTA for removal of heavy metals, sulfides, and other contaminants. And you need very long dialysis times, around 16 to 24 hours, with at least 2 to 3 buffer changes to get really salt removal and other removal of contaminants. They are more prone to leakage, especially at the ends where you need the closures to close them, to seal them. And if you use very long dialyzed tubings, you can, when you steer them, it could happen that they get small leakages because they might be in contact with the steerer and get damaged. You have a significant sample volume change due to more osmotic pressure, and you can't really handle small volumes, and they are not very suitable for high throughput analysis. That means for the simultaneous dialysis of a high number of samples, which might be interesting for screening experiments and other applications. And if you want to monitor your dialysis or remove samples for other tests during the dialysis, this is very laborious as well. This was a reason why ready-to-use dialysis devices were designed, which are much easier to handle, and they allow for dialysis of small sample volumes with only minimal sample dilution, and they are not so prone to leakage and other disadvantages with the standard dialyzed tubings. I want to show you two systems today. One is the Dialyzer system, and the other one is from the German company SERVA, developed new innovative system of dialyzing devices, the Express, Micro and Mini dialyzers. The Dialyzer system have ultra pure regenerated cellulose membrane pieces pre-installed in plastic vials of different sizes. The pre-treatment time is significantly reduced to 5 minutes, and for standard applications like salt removal, a 3-hour dialysis time is normally enough. You can easily open and close them with a screw cap. So that means that the sample loading and removal, and as well removal of samples during dialysis, is very easily done. You can load them with pipettes and don't have to use needles or other things that might puncture the dialysis membrane and then get as well some problems with leakage. So in principle, that's a leakage-free system, and you have a very high recovery rate of 98%. For dialysis in beakers or in other class vials where you need higher, larger buffer volumes, you can use a flotation ring for simultaneous dialysis of several samples, and as a buoyancy aid. They are available for different volume sizes of 10 to 150 microliter, 50 to 800, and 0.1 to 3 ml, but as well for larger volumes of 10 to 20 milliliter, and in different molecular weight cutoffs. Whereas the Dialyzer systems are more modification of the standard dialyzing tubing, the SERVA Express, Micro and Mini dialyzer systems are really a newly developed innovative system. They have a unique geometry which guarantees a very high membrane surface area to volume ratio, which means you have very short diffusion pathway, and this allows for extreme fast dialysis of as well very small sample volumes and with a very small volume change. In this case, there is no pre-treatment anymore necessary. They are of course leakage-free, and the sample recovery is even for small sample volumes up to 98%. And despite being a very new, totally different new system, you can load the samples and retrieve the samples with your standard micro pipettes. So again, you don't have to use needles or other things which might as well puncture the dialysis membrane, and you don't need any special devices for loading and retrieval. You just load the sample through the round opening at the top of the device, which is shown here in the slide. And it's very easy as well to retrieve it through this round opening again. They are moreover compatible with microtiter plates. So this is the first system for dialysis which can as well be used for high throughput applications. But if you want to dialyze in a beaker, or you need bigger buffer volumes because for some special applications, you can also use a flotation ring and use the dialysis in a beaker. The microdialyzers are available in volume sizes of 10 to 100 microliters and 50 to 300 microliters. You can use the single dialyzers in 2 ml microtubes, or you can use cartridges of 8 connected microdialyzers which fit perfectly in a 96 well microtiter plate. So this means that you can easily dialyze and as well change buffer for many samples in a very fast and easy way. You can also break away one single segment of the cartridge. So even if you use the cartridges, if you only use a single dialyzer, you can just break one away and make your single dialysis steps. And they are available as well in the molecular weight cutoffs of 3.5 kilodalton, 6 to 8, and 12 to 14 kilodalton. The mini dialyzer system is similar designed like the micro dialyzer systems, but they are for volume sizes of 150 to 1000 microliters. Again, you can use single dialyzer devices in a 25 milliliter tube, or you can easily couple them to a strip by a key slot mechanism, and then use again them in a microtiter format, in this case a 48 well microtiter plate, and again you can very easy dialyze and make buffer exchange for 48 samples at one time. To show you what I mean with a significantly reduced dialysis time with the micro and mini dialyzer systems, I want to show you two application examples. One is the removal of salt and one is removal of detergent. In the first example, there was the dialyzed sample with 100 microliter phosphate buffer against 1.8 milliliter water, and buffer was exchanged every 15 minutes. And then osmolarity of the sample was measured. After 60 minutes, you see in the chart, there's already a significant reduction of osmolarity in the sample. That means there's only a very low level of salt, residual salt in the sample. Even if your application tolerates a higher salt concentration, you can even stop, in this case, the dialysis after 30 minutes or 15 minutes. So this is a very short dialysis time compared to 16 to 24 hours with dialysis tubings. Removal of detergents like chaps or SDS is as well very effective and very fast. Especially detergents can really disturb downstream applications like mass spectrometry or as well gel chromatography or other things. So in this case, there was a 100 microliter BSA solution containing 1% chaps dialyzed against 1.8 ml water. And the whole dialysis time was 8 hours, and every 2 hours the buffer was exchanged. And you see again in the chart that after 8 hours, there's only 2.5% residual chaps concentration in your sample. That means after 8 hours, we reduced the concentration of chaps from 1% to 0.025%. That means a very, very low amount of detergent, which will not disturb any applications. Again, if you can tolerate a higher chaps concentrations, you can stop dialysis after 4 or 5 hours. And as a reminder, if we use standard dialyzed tubings, then we talk again about 16, 80 or 24 hours dialysis time. So in summary, the ready-to-use dialysis devices are not only much easier to handle, they as well shorten the hands-on time and the dialysis time significantly. They allow you not only to dialyze very small sample volumes, but in the case of the micro and mini dialyzer systems, you as well have the first time the possibility to use dialysis as well in a high throughput application. I hope that I could give you some interesting information about dialyzing, and I thank you very much for your attention.